boys. We will talk today male reproductive system. Male reproductive system consisting of a pair of testes. Now no more talk. Okay. Pair of testes, genital ducts, accessory glands, and copulatory organs. All these are come under the head of male reproductive system. Testes. Just below the testes, you are seeing this is the part of mediastinum testis, epididymis, and different ductules, vas deferens. So all these, first of all, see the testes. They are situated outside the body in the scrotum. No doubt they develop in the abdomen, and as they develop, they descend down and comes in the part of scrotum, where the temperature is about 2 to 3 degrees centigrade less than the body temperature. The reason is because this temperature is essential for the spermatogenesis. So because of that, it descends in the scrotum. Alright, the testes are covered by the 3 layers. The 3 layers are covering the testes. Outermost, tunica vaginalis. Tunica vaginalis is the serous sac which derives from the peritoneum. As the testes develops in the abdomen, they are surrounded by the peritoneum, and that peritoneum descending down along with the testes and comes in the part of scrotum. So outer layer, which is forming the serous sac, tunica vaginalis, is the derivative of peritoneum. It comes down along with the testes in the scrotum, covering the testes on the anterolateral aspect, and on the posterolateral aspect it is very diminished. Some books are mentioning absent, but it is very diminished on the posterior aspect. Next to the tunica vaginalis, there is the tunica albuginea. Tunica albuginea is a part of dense connective tissue, and that dense connective tissue covering the posterolateral anterolateral aspect of the testes, and on the posterior side of the testes it is thickened, and this thickened part of the testes is called mediastinum of the testes. From the mediastinum of the testes, the different septa are passing inside the testes. You are seeing here one lobule, second lobule, third, like this. So many lobules are formed by the septa which are coming from the mediastinum of the testes. What is the mediastinum of the testes? That is the thickened part on the posterior side of the testes. Tunica albuginea is thickened, and that thickened part is called mediastinum. From this, the septa are passing inwards inside the lobule, and they are dividing the testes into large number of the lobules. These lobules, you can see in this diagram here, one lobule, second, like this. So many lobules are formed by the septa coming from mediastinum of the testes. Alright, now in each septa or in each lobule, what we find, there is seminiferous tubules are present. The number of the seminiferous tubules in each lobule, you are seeing these circles, coiled structures, these are the seminiferous tubules. So each lobule is containing about 1 to 4 seminiferous tubules. Alright. Surrounding the seminiferous tubules, I am talking of one lobule. Surrounding the seminiferous tubule, there is present the interlobular connective tissue, or this connective tissue is also containing the another type of cells. That cell is the Leydig cells, which is secreting the testosterone. So when we see the lobule, each lobule is having 1 to 4 seminiferous tubule. One thing. Surrounding the seminiferous tubule, there is present the interlobular connective tissue mass. This connective tissue mass, no doubt, it is also having the blood supply, and that is the tunica vasculosa, rich in blood supply. That is also present there. And besides this, there is present the Leydig cells. So where the Leydig cells are present? In the interlobular connective tissue mass. So these are the structure of each lobule. From the seminiferous tubules, the efferent ducts comes out, and all these efferent ducts, they join together here. You are seeing the structure here. Straight line is lobules are here, straight line is lobules are here. So from each lobule, each straight line comes here in the mediastinum, and they join together, and this bunch is called retitestes. You are seeing this bunch. How this bunch is formed? This is formed by the straight tubules coming from each seminiferous tubules, and they join together here and forming this bunch, and this bunch is called the retitestes. From the retitestes, again you are seeing the straight line. दिख रही है आपको? These straight lines, they join here with the head of the epidermis. ये called this C-shaped structure, larger structure which is present on the posterior side of the testes. This is called epidermis. So very simple thing. Each testis is surrounding by tunica vaginalis. First thing, that is the part of peritoneum. Next, tunica albuginea, that is thickened on the posterior side. You will read it again in the gross part. I am telling you this because without this understanding, you can't understand the another thing. Then, tunica albuginea, which is thickened on the posterior side, that is called the part of epidermis. From this, the septa are passing inwards. All right. Each lobule is having the 1 to 4 seminiferous tubules. So these straight lines, they join together here and forming the bunch in the part of epidermis, that in the part of mediastinum, not the epidermis. Epidermis is here. That is called retitestes. From the retitestes, the straight line join together to this C-shaped structure on the posterior side of the testes, that is epidermis, head of the epidermis. From this head of the epidermis, it is very coiled structure, single coiled structure. It is continued downward into the another coiled tube, and that is the vas deferens. And this vas deferens, the distal part of the vas deferens, see here, it is dilated, that is called ampulla. And below the ampulla, here is the one gland, another gland is surrounding the urethra. This is the prostate gland, and this is the seminal vesicle. So the duct of the seminal vesicle join with the ampulla, that is duct of the vas deferens, and the combination of the two is named as ejaculatory duct, and this ejaculatory duct opens into the urethra, which is present inside the prostate gland. As it is lying in the prostate gland, that urethra is called prostatic urethra. I will use this word again because the epithelium will change in prostatic urethra and penile part of the urethra. Okay. So this is the ejaculatory duct. Ejaculatory duct open into the urethra, and as we go down, this is the bulbourethral gland, and rest of the part is penile urethra. As I used the word previously, a pair of male reproductive system consisting of pair of testes and the genital ducts. See what are the different part of the genital duct that I am telling you. The genital duct includes, what is this? Straight line which are coming from retitestes to this. Efferent ductules. This is the efferent ductules. I am talking of these straight tubules which are coming from the retitestes and are reaching to the head of epidermis. These are the efferent ductules. So one is the efferent ductules, then the epidermis, then comes the vas deferens, and then part of the male urethra. All these are the parts of genital ducts. Besides this, the third part of male reproductive system, that is of accessory glands. The accessory glands include, first is the prostate gland, this one. Another is seminal vesicle. Third is bulbourethral glands. So all these are the accessory glands of male reproductive system. Alright, before coming to the testes, I just want to tell you about the glands. Prostate gland. The prostate gland is secreting the secretion, prostatic secretions. All the three glands, they are secreting the secretions, and that secretions help provide the movement of the sperms, one thing. Also provide the nutrition and this thing. The part prostate gland is having the urethra that I will tell you just now. Prostatic urethra, that epithelium is transitional epithelium. That is important because once you identify the slide of prostate gland, the examiner will ask you what is the epithelium lining the prostatic urethra. That is transitional. In rest of the part, that is the part of the stratified squamous non-keratinoids. Alright. Now see the part of testes. The testes is the part of the male reproductive gland which is provide the sperms. And the different stages you will see in the part of testes. When we focus the slide of testes, you will find the such rounded structures. All these are the seminiferous tubules. ये दिख रहे हैं आपको बड़े? These are the seminiferous tubules. Lying between the seminiferous tubules, there is present the connective tissue mass. In the connective tissue mass, you are seeing this small group of cells, bunch of cells, Leydig cells. And what is the other thing present in this connective tissue? Blood vessels. And what is the supply? The innermost covering of the testes is tunica vasculosa. Tunica vasculosa is rich in the blood supply, and that is present in the connective tissue mass. So these three things, tunica vasculosa, Leydig cells, and also the connective tissue mass, all these are present between the seminiferous tubule. Clear it? Now see the seminiferous tubule. See this diagram. This is the seminiferous tubules. The seminiferous tubules is having the two type of cells. One is the spermatogenic cells, another is long Sertoli cells. Look this diagram. You are seeing the elongated cells. These elongated cells having the nuclei, elongated nucleus. This is called Sertoli cells. The Sertoli cells, they are providing the nutrition and support to the spermatogenic cells. Position क्या है इनकी? Between the spermatogenic cells or not? Look here. This is the spermatogenic cells. Spermatogenic cells. Between them, you are seeing the elongated cells extending from basement membrane to the lumen. These are the Sertoli cells. Sertoli cells, they provide the nutrition and support to the spermatogenic cell. One. They are also acting as a phagocytic. That means excess of cytoplasm is formed that has been removed by Sertoli cell. Besides this, the important part of Sertoli cell, they are secreting the hormone, androgen binding protein first, and second hormone inhibin. The androgen ABP, androgen binding protein, it concentrates the testosterone and also absorb the excess of fluid from this part. The second, inhibin, this hormone acting over the FSH, you will read, that is the part of suprarenal gland that we will talk later on. It is acting over the FSH, follicular stimulating hormone, to inhibit the secretion of androgen binding protein, which is coming from FSH. So that is also the function of Sertoli cell. Next to the Sertoli cell, you are seeing this bunch of the cell starting from the basement membrane. Lying over the basement membrane, this group of cells is the spermatogenic cells. So I told you that the testis is consisting of two types of cells. One is the Sertoli cell, another is spermatogenic cells. The spermatogenic cells lying over the basement membrane, that means this part, they are the immature cells. They are the immature cells and they come from the yolk sac to the testes since in the beginning, 3rd to 4th week, and remaining in the testes up to the puberty. One thing. Just after the puberty, so the cells which are lying over the basement membrane, these are the immobile or they are not acting as a spermatogenic cells. These cells later on, at the time of puberty, they undergo the mitotic division. By this mitotic division, they forming the primary spermatocytes, and this primary spermatocyte is having the diploid number of the chromosomes. Now, later on, this primary spermatocytes, you are seeing the second cell, primary spermatocytes, it undergoes the first meiotic division. It goes under the first meiotic division and forming the haploid number of the secondary spermatocytes. And this secondary spermatocytes again undergoes the second meiotic division and forming the spermatids. So from the primary spermatocytes to the formation of the spermatids, that is the stage you have read in the embryo. First stage of spermatogenesis. First stage of spermatogenesis. So we have reached up to the part of spermatids. These spermatids are immobile. These are immobile. Undergoes the first meiosis. First meiosis division, spermiogenesis, and by this spermiogenesis they become the motile, and this is the stage up to the then undergoes the second spermiogenesis, or you can say or try to understand when the immobile spermatids become mobile sperms. This is the completion of the second spermatogenesis stage, and this mobile sperms, they develop after the second meiotic division and they are resting here. So when we see that slide of the testes, you will find all these structures in the seminiferous tubules. Lying over the basement membrane, the spermatogonium, primary spermatocytes, secondary spermatocytes, ultimately in the lumen. Lumen is filled with the sperms. Alright? But it is not sufficient. Look here, this is the one seminiferous tubule. This is another seminiferous tubule. This is another like this. My point is that the interlobular connective tissue mass between all these seminiferous tubules, this is the interlobular connective tissue mass. This interlobular connective tissue mass is showing this group or bunch of the cells. These are the Leydig cells. So Leydig cells are present in the interlobular connective tissue mass, and these Leydig cells are releasing the hormone testosterone. So all these structures you will see in the transverse section of testes. Any doubt? Okay. Next slide, you will see the epididymis. Epididymis, as I shown you the gross structure, like this. It is a C-shaped structure, highly convoluted single tube. A large number of the convoluted mass and single tube starting from or present on the posterolateral side of the testes. It is having the head, neck and tail, different parts that you will read in the gloss. When we take the transverse section of the epididymis, the transverse section of the epididymis is showing the three balls, mucosa, muscular ball and outer covering. The epididymis is lined by, you are seeing this large number of the circles. These circles, the one thing, they are lined by the pseudostratified columnar epithelium. Alright, one, pseudostratified columnar epithelium. But you will also find the pseudostratified columnar epithelium in the trachea, also some part of the respiratory system. How you will identify? Alright, you identify the pseudostratified epithelium. The lumen of trachea is showing nothing. That is showing the hyaline cartilage. No cartilage you will see here. Okay. No cartilage seen here. Another thing, surrounding the lobule, you are seeing the completely encircled smooth muscle fiber, which are not seen in the trachea. Follow my point. In trachea, no doubt you will see the such structure, large number of the circuits lined by the pseudostratified columnar epithelium, but they are not surrounded by smooth muscle fiber. Here, each one is surrounding with the smooth muscle fiber. The role of smooth muscle fiber, their contraction help in releasing the sperms. Third point, see the lumen. Lumen is very much filled with the sperms. Lumen is filled up with the sperm. So we can easily differentiate that this is the slide of epididymis. I want to tell you here, once you see the pseudostratified epithelium in any circle, don't immediately say that this is this. Confirm yourself whether the slide is epididymis or slide is of trachea. I have told you the points, differentiating points, how you differentiate. So see those points and then speak, then give your diagnosis, not before. Another point, the function of the epididymis. One is storage of the sperms. Also give the passage for the maturation of the sperms. Third, it is also provide the nutrition to the sperms. That is the part of epididymis. Next, ejaculatory duct, the slide of vas deferens. In previous classes, I have told you what type of the lumen you are seeing here. Bolo. And where you find the star-shaped lumen beside this? आपसे पूछ रहा हूँ खड़े हुए. Besides star-shaped lumen seen here, where in other slide you have seen the star-shaped lumen. हाँ? तो फिर पहले आप यूरिथ्रा क्यों बोल रहे हो? हैं? अगर ये अभी कंडीशन ये है तो एग्जाम तक क्या कंडीशन होगी? बैठ जाओ. You also see the star-shaped lumen in the part of ureter. So once you see the star-shaped lumen, your mind should click. The slide can either be ureter or of the vas deferens. Then we must know the differentiating point. The differentiating point, see the muscle coat. Muscle coat is same. Inner longitudinal, outer longitudinal, middle circular. The same muscle coat you find in the ureter. So only one point is left. Which part of the ureter is having the 3 layers of muscle coat? खड़े होकर बोला करो सरदार जी, बैठ जाओ, ठीक है, सही है. In the lower 1/3 part of ureter, okay, only the part differentiating point left is epithelium. So confirm yourself, epithelium is transitional or pseudo-stratified, and that confirmation you can do under the 40x. Mind it. So all of this I am telling you because once you see in the slide star-shaped lumen, differentiate yourself. And all the points I have told you that how you differentiate. So that is the vast difference. You see this diagram also. Star-shaped lumen, inner longitudinal lamina propria part, inner longitudinal muscle, circular, middle circular, and outer longitudinal muscle cord. Then I come to the part of accessory glands. In accessory glands, one is the prostate gland. The prostate gland is consisting of large number of tubulo-alveolar glands. You understand my point? The glands which are present here is having the shape tubulo-alveolar, means upper part is tubule shape and lower end is circular. So that is called tubulo-alveolar glands. And these tubulo-alveolar glands, large number are present. Look here, some are tubule shaped, some are alveoli. So these tubulo-alveolar glands, the lining of the alveoli depends on the activity of the alveoli. If the alveoli are active, the epithelium may be columnar. Listen carefully, otherwise you will remain confused. If the gland is not active, then the alveoli will line by cuboidal epithelium. So my point is, the prostate gland alveoli may be showing lining of columnar cells, or it may show the lining of cuboidal cells. Okay, so don't go any doubt. Sir, इसमें cuboidal दिख रही है. All right. The stage is inactive stage. If it is showing the columnar epithelium, that means active stage. Right? The third thing, you will also see the section may include the part of prostatic urethra. So, if the prostatic urethra is present, it is showing the transitional epithelium. But it is not necessary that the section includes the prostatic urethra, or yes or no. Depending upon if the section is including the prostatic urethra, then you can see the prostatic urethra in the prostate gland. If it is not including, you will not see. But in prostate gland, you will always see the large number of the alveoli, maybe the tubular shape or alveolar shape. These were always present. And another thing, in the alveoli, you will find the secretion mass. You are seeing such type of secretive masses are present. These secretions are the prostatic secretion or prostatic concretion. These are present inside the part of each alveoli. The function of prostatic secretion, they maintain the nutrition of the sperms and also provide the media for the movement of the sperms, as that is common for all the glands. The glands are three types, and these glands secreting the secretions, that secretion providing the media for the movement of the sperm, also provide the nutrition. So that is the part of prostatic glands. Another gland, the seminal vesicle, which is, I am not giving you slide of seminal vesicle. You just keep one thing in your mind. it is also showing the large number of the alveoli, and those alveoli are like the honeycomb appearance. Their epithelium is also columnar. The important part that they are secreting the secretion, and that secretion is alkaline in nature, having the fructose, and that fructose is necessary or providing nutrition to the sperms. Bulbourethral glands, which is lying surrounding the part of the urethra, male urethra just below the prostate gland, that is also providing the secretion. After this, then we come to the part of copulatory organs, penis, that is showing the three erectile mass. It is consisting of three erectile mass. The posterior one, these are the corpus cavernosa, and anterior, that is the corpus spongiosum. The corpora cavernosa are present on the dorsal surface, and corpus spongiosum is present on the ventral surface. Look, this erectile mass is surrounded by the large number of the dense connective tissue fibers and this. In the interior of each corpora cavernosa, you will find there is present the deep artery. You are seeing the circle? This is the deep artery of penis. And in the corpus spongiosum, there is present the part of the urethra. And this urethra is lined by transitional epithelium. The urethra present in the corpus spongium part, that is lined by the transitional epithelium. Oh, sorry. This is lined by the part of pseudostratified epithelium. Then we come to the part of, see the dorsal surface over the covering. You are seeing here the another artery, that is the dorsal artery of the penis, and nearby it there is the dorsal vein. So all these structures, they are seen in the penis. So these are the slides which you will see in the part of histology, that is complete of the male reproductive system. Look this diagram. When you focus the testes, you will see this picture. Seminiferous tubules, radix, and in the each one you must know the Sartoli cells and all this. That is the part of male reproductive system. Okay. In the next class, I will complete the female reproductive system, then endocrines. Along the endocrines, I will tell about the skin also. In the first week of August, I will finish this. And I am telling you previously, in the second week of August, I will have the part completion test, including the complete abdomen. That I am giving you advantage to the students who are below the pass marks up to the second semester. So cover this, because in the last of the August we will have only one exam, PUC exam. If the marks of you, anyone who is having not pass marks in the PUC cannot appear in the university exam, take it granted. And we will not help you. Reason is because everything is going to the NMC. So keep this thing in your mind. फिर मत बोलना सर हमें पास कर दी. I can't. So I am take it either warning or take it as advice. Start reading and maintain this. That's all. I am seeing the strength of the student is diminishing. Why? What is the reason behind? They don't want to appear in the exam? Ask your friends, what is the problem? Give me paper, small paper. I take the attendance because I have the time.